Hello, everybody, and welcome to the virtual TrekCon. Our panel here today is called Barclay's Protomorphosis Syndrome and the Viruses of Star Trek. Very timely. So let's start. Dr. Lin, could you introduce yourself, please?
Hi, I'm Audrey Lin, and I'm a postdoc at the National Museum of Natural History in Washington, D.C. And I'm an evolutionary biologist. I study ancient biomolecules, for example, DNA that had been isolated from bones, teeth, and the tissue of animals like birds, dogs, and pigs, and bats. And sometimes these animals are thousands of years old. And I use ancient DNA to try to answer questions about the evolutionary history of these animals. And I'm also interested in the evolution of zoonotic viruses, like what are the characteristics that make them more likely to switch host species and to establish themselves into new hosts.
Awesome. Thank you. That's fantastic. And that's in the Smithsonian Institution. Is that correct?
Yeah, yeah.
Excellent. Dr. Duplessis.
Hi, I'm Louis. I'm a postdoc in the zoology department at Oxford, and I'm also an evolutionary biologist, but what I do is genomic epidemiology, which means that we sample virus or other pathogen genomes from infected patients, and then we look at genetic changes in those genomes. We look at the mutations to reconstruct the evolutionary history of the disease. So we can model the spread of the disease, and we can look back in time to before the first infected case.
That's fantastic. Well, I'm Mohammed Noor. I'm a biology professor at Duke University, and I'm an occasional science consultant for the Star Trek universe. I'm also an evolutionary biologist, like my esteemed colleagues here, and my research is on the genetic changes that lead to new species formation, primarily using the fruit flies Drosophila. But we're not talking about Drosophila here. We're going to be talking about viruses, and let's open with a Voyager episode, Macrocosm. Louis, would you mind giving us a quick intro to that episode?
Yeah, so I want to take you to close to the end of the episode, actually. And towards the end, Captain Janeway is making her way through deserted corridors of the Starship Voyager towards the holodeck. And she's got her phaser rifle and she's got an antigen bomb, which is going to use to destroy the giant viruses that have taken over the ship. And just as she's about to detonate the bomb, she gets ambushed by a giant virus that tries to stab her with its spike protein. And then she manages to wrestle the virus off her and then pull out a knife and stab the virus to death. And I think that scene is just a bit cathartic because it's a bit of a revenge fantasy to be able to stab a virus and physically attack it, because viruses are so, they're so tiny, you can't see them, and you don't even know if you're infected for the first few days. So even though it would be really scary to have these giant viruses attack you, it's just might be nicer to be able to see your enemy. So that's why I thought that scene would be great to start us out with.
That's outstanding. So how big is a typical virus? And like, are there some really big, I mean, obviously not as big as the macrocosm ones, but are there some big ones that people may have heard of?
So a typical virus is about 30 to 50 nanometers across, and that's really, really tiny. So I was going to say like a human hair is like 30,000 nanometers across or something like that, but that doesn't really help us. So if you were to imagine that you were the size of a human cell, then in comparison, a virus would be like a really small mosquito. It's like we don't really realize how much smaller viruses are than human cells, or even smaller than bacteria. They're really tiny. And like when they were first discovered, people didn't, there was nothing you could do to actually observe a virus. Like you can't see them through a microscope. You need an electron microscope, which hadn't been discovered at the time. So you had to observe them indirectly by just looking at their effects. Yeah, I'll hand over to Audrey to talk us through the really big viruses.
So there are some really, really big viruses that are so big that they can be even bigger than certain types of bacteria. And they're so big that they are even infected by their own viruses. And these are known as well, they're called Mimi viruses, megaviruses, and Pandora viruses. And I think that they were first discovered in an air conditioning unit or like a water tower in Marseille. So these scientists just decided to see what was in there, and they found these new organisms that had never been, their existence had never been known to us before. Another kind of famous giant virus is the Pythovirus sibericus, which is a 30,000-year-old giant virus that was found in the permafrost in Siberia. And this virus is 1.5 microns across and most likely infected amoeba.
So I have to ask on the permafrost one, is that one that can come back to life, or is it just there as a husk?
No, it's
Louis.
infectious, right? They revived it and they used it to infect amoeba.
Wow.
Yeah,
Wow.
but that's a special case because those giant viruses are actually quite hardy compared to most viruses.
So it survived 30,000 years in the permafrost. Wow.
If I can interject quickly, we only discovered these giant viruses fairly recently because no one was looking for them, because we thought viruses were really, really tiny. And we never thought to look for giant viruses.
That's fantastic. So back to the episode, the virus that was attacking Janeway had this big spike. It was trying to like puncture her in there. So a very dramatic scene. And I've heard of something called spike proteins associated with viruses. Is that a spike like that, or is that just a name like that?
So they can be called spike proteins, and they're actually cell surface receptors that are on the surface of the virus cells. And they don't exactly, generally, they don't exactly stab the host cell, but it functions more like a lock and key mechanism. But it's a bit more complicated for other potential host cells like bacteria and plants that have a much more complex cell wall than mammals do. So I guess an example of a virus that does have a kind of spike protein would be a bacteriophage, which we will be talking later. But they have spike proteins that bind to the receptors of the bacterium and then work as a drill of sorts, kind of cleaving away a hole as it goes. And in any case, the way generally that a virus enters a host cell is binding to the cell receptor on the host cell, like a lock and key, and then fusing with the host cell membrane, and then it dumps its contents, like its genome, into the host cell.
Basically makes the host cell into its slave to make more viruses, I assume, then.
Yeah, exactly.
It's so evil. So in this episode, I have sort of two related questions. This episode, we have these viruses are just kind of flying around. Now we hear about like airborne viruses, but I'm assuming that like if you're a virus that big, you couldn't just fly. I guess a more real scientific question is, do viruses, are they like attracted to specific targets, or do they just float around till they run into something?
No, viruses can't move around by themselves. They don't have any mechanism to make them move around. They don't have any agency either. A virus is just it's a mechanical object really, and it just floats around. And so when they say airborne viruses, those viruses are usually caught in little water droplets that evaporate, and then they're carried around by air currents around the room or wherever. So that's why I guess good ventilation is really important.
That's excellent. So in terms of the resolution of the episode, there was a lot of references to antigens. Can you define, please, what an antigen and an antibody are? Because those come up a lot in Star Trek in general, as well as in this episode.
So yeah, antigens don't destroy viruses by themselves. Antigen is basically any molecule that the host immune system recognizes as non-self, something to attack. So in this context, then an antigen is a molecule that's produced by the virus. There's no need to synthesize an antigen. And antibodies are Y-shaped molecules that had been secreted by immune cells called B cells. So antigens, in this case, are bits and pieces of the virus that the antibody specifically recognizes. And then there's another lock and key mechanism, which is kind of a ongoing theme here. And the virus is then marked for the immune system, like a red X, to be destroyed by the immune system. And B cells can also bind to the virus and destroy the virus.
So injecting the virus with antigen like the doctor did earlier in this episode would have not served any purpose at all.
Well, it's a good segue. So what would have made this episode a little bit more accurate, do you think?
So basically, if it were any other kind of organism besides the virus, like
something multicellular, really.
Yeah, like a plasmodium or a helminth that went through a really dramatic metamorphosis.
I was thinking it looked a little bit like bot flies, which are parasitoids, where they'll lay their eggs inside a person and then the offspring will emerge. It was a little bit like those bot flies.
They're really disgusting.
They
don't
are
Google
disgusting.
that.
Yeah, no, you don't want to. But that may have worked actually pretty well, I guess.
Yeah, the other thing I thought would have been good is if the doctor had actually just sterilized his mobile emitter when he beamed back onto Voyager. Would have prevented everything.
Yeah, I think wasn't there a comment in there about how like they did it, but they took them a couple of extra minutes or something. Like he said something after he beamed back.
So they didn't have this virus in the data bank, so they couldn't scrub it from the transporter.
Yeah.
But he could have just beamed directly into some UV light or into a chlorine bath or something like that.
That's true, and it's not like the doctor would have gotten hurt. Good point. Okay, any final comments on this episode before we move on to the next one?
I'm good.
It was a fun episode to watch, especially for Janeway.
Definitely.
It's the badass Janeway episode.
Definitely. She reminded me a little bit of Ripley from Aliens when she was wandering around with the big gun.
Yeah, definitely. Yeah.
All right, well, let's move on to our second episode. This is an infamous one. This is from Star Trek The Next Generation, the episode Genesis. Louis, do you want to walk us through? Is it Louis or is it no? Audrey.
I'll just start it out and say that this episode kind of it's got something which is really reminiscent of Macrocosm, which is in Macrocosm, Janeway and Neelix go back to Voyager and find the ship adrift, and with all the crew missing, and that's kind of like what happens in Genesis. Isn't that right, Audrey?
Yeah. So I guess this is another one of the another body horror episode that Star Trek likes to do periodically. And yeah, it's like generally not very well regarded for its scientific accuracy, but it does feature some of my favorite scenes. Such as Barclay, who is one of my favorite characters, de-evolving into a giant spider and building a nest in the warp core. Spot giving birth to a litter of kittens and then turning into an iguana. And Worf storming into Troy's room, I guess off camera, and biting her in the face. And Riker regressing into a Australopithecus. Very, very
He's regressing and he keeps forgetting things. That's kind of how I feel a lot these days.
Well, this was interesting in the sense it brought up a lot of both evolutionary concepts in the sense, I'll give them a shout out. They at least acknowledge the common ancestry of all species on Earth there. So shout out to that. That's not universal. Yeah, it's not universal. On the genetic side, there was an interesting comment near the beginning where Dr. Crusher said something like you have one dormant gene out of 100,000. And right now, the estimates of the number of genes we have is on the order of like 20 to 25,000. But if you go back, I remember when I was working in a molecular biology lab in 1996, I remember people tossing out this 100,000 number because no organism had all its genes sequenced yet until I think it was 1996 was the first in terms of either a fungus, plant, or animal, and that was the yeast. So kudos, they at least got the number accurate for the time. So 2 thumbs up to them for that.
And I think now we know that genes only make up about 1% of the human genome.
Definitely. So Audrey, do you want to explain what an endogenous retrovirus is? Because I think that came up in this context.
Yeah, so this topic actually is kind of is a little dear to my heart because I worked on this during my PhD, but essentially endogenous retroviruses are ancient remnants of ancient retroviral infections that make up to 8% of the human genome, which is a huge amount. And basically how they got in there, so part of the retrovirus life cycle is to incorporate its genome into the host DNA. And these endogenous retroviruses had originally infected a sperm or egg cell of one of our very distant ancestors. And then every time that cell would divide, there would be a copy of that retrovirus in the new cell. So most of these endogenous retroviruses have been deactivated over time because, I mean, millions of years have passed. They don't just, and for the most part, they don't do anything. They just sit there in our genome. But there are certain instances where the host has co-opted the retroviral DNA for our own purposes. Like the protein syncytin was taken from the envelope gene of the retrovirus, and it's used for the formation of placenta. And which is interesting given that pregnancy is a big part in this episode. And this biological property is so important that it even, that this happened independently more than once in different common ancestors of mammalian species. And also these endogenous retroviruses are involved, some of them are involved in other important processes too, like innate immune function and embryonic development, the formation of blood cells and starch digestion as well.
That's awesome. That's really cool that basically a big fundamental part of us as, a big section of mammals we actually acquired from viruses.
Yeah.
That's really cool.
Definitely. So we all have viral ancestry.
That's amazing.
Do you know if anyone knew about ERV's endogenous retroviruses back when this episode was written?
I don't know.
In 1995.
Yeah, I don't know.
1994, I think.
Yeah, I don't know the answer to that. It's a great question though. So, go ahead.
Oh, sorry. I think that the science that there are researchers that we did,
Wow.
but probably the writers.
It's fair. They're not like in-field biologists. So there's a lot of good things about this episode that we highlight, but there's clearly some broad confusion. Like Dr. Crusher mentioned something about the T cell in your DNA. I don't think that even makes sense, does it?
Yeah. Like that was one of the things which I kind of was trying to look past that, but I just
I think they
So-
just inserted words for the technobabble.
Yeah, it's the biology equivalent of quantum. They always do with physics. When something's cool, add the word quantum to it. So biology will add T cell.
Retrovirus.
Retrovirus. The other thing which was problematic there was the use of the word intron, right? So they introduced, they said that, or Data said to Picard that the introns were activated, and that's what caused it. Now, DNA guides what amino acids are put together to make proteins. And the stretch of DNA that does that is what's called a gene. Everybody's heard of genes. And within some genes and some species, there's sections of the DNA that don't actually have information about the amino acids in the eventual protein. And those stretches are what's called introns. So I don't need to go into the mechanics of what happens with them, but that's basically the punchline. But these are not, quote, evolutionary holdovers, like Mr. Data said. Like there's no way activating an intron would make anything even remotely like an ancestor. However, what I think they were going for was something called a pseudogene. The pseudogenes are genes that are in present day species, but they had a function in an ancestor that they no longer have today. So a great example of a pseudogene is the vitamin C gene in humans. We all have the gene for producing vitamin C, but we obviously don't make vitamin C ourselves. We have to take supplemental vitamin C or take it through our food and things like that. At some point in time in the past, some mutations broke up that gene. We still have it, but it doesn't work. It's not turned on. Now, the problem with this, even with pseudogenes, is even if you turn those on, it's not like you'll suddenly just be making functional vitamin C because it's acquired mutations over time while it wasn't functional. So even if you actually fix the term, it doesn't actually fix the fundamental problem, at least with that one.
Yeah, you don't carry your ancestors' genes inside your genome.
No.
In a way you do, but you're still using most of them actually.
Exactly. Because we share we share a huge fraction of our genes with just about every, animal species and even many other species that aren't animals. Louis, Dr. Crusher named the syndrome after Lieutenant Barclay. Is it normal to name syndromes after patients? And can you explain how this would work today?
I think it was much more common, but so the current thinking is that you should never name a disease after a person or a place, especially not a person or a place that had the disease. So sometimes they are named after the discoverer of the virus, like the Epstein-Barr virus, which is not named after any Epsteins or Barrs you might know. But for instance, so back in the day,
like Legionnaire's disease, I guess, is one which
Legionnaire's disease.
Yeah.
The one I was going to bring up is syphilis, which was called the French disease by the Italians and the Germans. Ooh. It was called the Spanish disease by the Dutch. It was called the Polish disease by the Russians, and it was called the Christian disease by the Turks. So it was really just a blame game, and everyone was blaming each other for that. And that's why you shouldn't do that, because, or why we try not to do it anymore. We're still doing it to some degree. I mean, the other famous one is the Spanish flu, which was named after Spain, which was not the first country to be infected.
Oh my goodness.
But we shouldn't do that, because if in this case, naming it after poor Lieutenant Barclay, it kind of passes the blame to him for starting this whole disease, and he's really not to blame for it.
No.
So we try to
call the Crusher disease at that point.
He did start it, actually.
So what do you think would have made the solution to this episode more realistic overall? It's a tough one.
So I think, well, if you're already at this point where everyone has devolved into their ancestral species, and now you have to get them back, simply injecting them with some sort of inoculant, I'm not sure what Data put in there, is not going to change anything, because they've already devolved and you can't just turn back the clock again. So what would have worked is if they had actually used their transporters. So in the transporters, they've got something which can reconstitute matter, but they also have a record of everyone's full genome, and they also have transporter records of the last time anyone beamed in. So they could just have reconstituted themselves to slightly before the whole episode started. I think that might have been a better resolution, or a more realistic resolution, than just trying to cure them from this disease magically in no time.
Yeah, that does open the question whether they could just make clones come out too, but that's okay.
That's not allowed.
We won't touch that one for now. Any final comments on this episode?
Fun to watch. kind of infuriating.
I wanted to make the observation that this episode is a testament to why Star Trek needs someone like you, Mohammed, on the writing team.
So actually, I could make a quick comment on that. The science consultant, I believe at the time, would have been Andre Barmatis, who's a physicist. He's trained as a physicist. And he said at various talks that when there was something more biological, he had a friend who was a doctor and he'd consult with them. But it's likely the doctor hadn't had an evolution class, in 15, 20 years or something like that. So some of the details, like specific terms and things like that, would have been tough. The other thing is, of course, it is science fiction in the sense that you don't get to say no, you just get to answer how. So sometimes you could be given an impossible problem, like, all right, I'm going to make up something and call it spore drive or whatever. Just kidding about that one. Anyway, on that quick note, I do love the spore drive, by the way. Let's move on to the third episode. This is from the first season of Deep Space Nine, called Babel. Audrey, could you give a quick recap of the overall plot of that one?
So in Babel, a virus, mysterious virus, infects the station's residents, which makes almost everybody, unable to speak coherently. And the virus is mimicking the effects of aphasia, which is the connection of words and meanings in the brain. And this connection is scrambled in those infected. And I liked this episode because there are many scenes in the beginning of a very grumpy, sleep-deprived O'Brien trying valiantly to fix a failing space station.
Awesome. So are there viruses that produce neurological problems like aphasia, and if somebody got them, would the effect be long term, like where they'd just be like they'd be broken, or would it be short term, where it actually can get fixed pretty quick?
So there aren't viruses that actually produce like the effect of aphasia, but there are a number of viruses that do affect the neurological function of those infected, like herpes, and now we're learning COVID, polio, rabies, a bunch of enteroviruses. Basically, these viruses that can cause inflammation, brain inflammation, or encephalitis. And in this episode, the aphasia virus seemed to originally be transmitted through infected food, and this is kind of similar to the polio virus, which has an oral fecal transmission route, and then migrates from the stomach to the lymphatic system, and then to the central nervous system, where it would cause paralysis. So a lot of these viruses that I had mentioned are they're long-term. There can be several years before these sorts of symptoms develop. Is there anything else that that Louis wants to chime in with?
Not really there. Yeah, I think it's just the question about long-term damage. I think in most cases the damage is irreversible, isn't that right?
Yeah.
So that was quite special about this episode, that it just somehow constricted the synapses or something. I don't remember exactly how they said it, how they explained this away.
It was stopping the action potentials. They didn't say that. I just made that up. Just kidding.
They said something about synapses.
Yeah, I think they did.
I believe neurons can't. Sorry, I believe that neurons can't be regenerated. So once they are killed, they're gone.
Oh wow.
They grow really, really slowly as well.
Gotcha. So in this episode, Dr. Bashir said something about the nucleotide sequence of this virus being "quote unquote" too perfect. What does that really mean? And could you actually distinguish an engineered virus from a natural virus in some way using the nucleotide sequence?
I think it would be difficult, but you could perhaps do it by comparing it to known viruses. So if we were to genetically engineer a virus, we would probably start out with something we know that we've used in the lab before. So if you find something that's very, very similar to something we know quite well, there's a good chance it was engineered. So yeah, we could look at that. So we use something like a backbone or a scaffold to maybe just change a small function there. But something we might also want to think about is with our current tools, which are not the ones they have in the 24th century, of course, we can engineer a virus, but our tools are very, very blunt. So what we might do now to engineer a virus is to just take something we know has some properties we want and just evolve it in the lab by running it on some competition experiments until it starts to gain some functions. So experimental evolution is a thing, and it's actually quite powerful.
That's awesome. Any other comments on that from Audrey, or is it?
Yeah, so I just wanted to add that I guess you can hypothetically design a synthetic virus. It's more common now for bacteria. People do design synthetic bacteria, and but what the synthetic microorganisms can do is really limited. And I suppose it is like possible to add signatures, like what the Bajoran doctor did in the episode in the virus genome, like Decon was here to be translated into acid sequences. This could be possible in very large viruses in which there's a lot of redundancy in the sequences, like
A large genome, not large physical size. Is that right?
Oh, I'm sorry. Large genomes. Yes. Like herpes viruses have genomes that are like on average 250,000 base pairs long, versus say an HIV genome, which is only 10,000. So
10,000?
more probable.
Wow.
Yes.
I didn't realize it was that tiny.
That's
like a human
really
gene.
Tiny.
That's like a human gene. That's
but
amazing.
Actually, they're so packed. I don't know if you've seen this. They've got overlapping genes, and in some places they've got 3 genes at the same place. So it's just crazy. They have no extra space for anything.
Wow. Wow. Okay, that's very dramatic. So, I mean, you guys mentioned a couple of the problematic aspects with this episode in the sense of, the long-term damage. What are some other problematic episodes with the depiction of this aphasia virus?
My biggest issue with this episode is actually just the total and complete lack of PPE. So
define what that is. Sorry, some people might not know.
Personal protective equipment. Yeah. So any sort of mask wearing or any sort of isolation wards or etc. There's virtually nothing of this in this episode, and that's actually not strange for Star Trek, because I think across all the Star Trek series, I don't think we ever see a doctor wearing a mask or a visor.
It's rare. It's rare.
It's very, very rare, and I don't know if that was a budgetary issue or if they just didn't think about it. But clearly, this would be like the first thing you would do is you would make a real isolation ward, and not the isolation ward they had, which was a joke. So you would really want to try to isolate.
Oh no!
Of course, because he's the most important person, and you also want to try to stop the virus spreading to parts of the station it hasn't reached yet.
Yeah.
Instead, they were just sort of like, oh, it's airborne.
We'll put them all in a room.
Everyone's infected.
Was not a great approach to that. So aside from having a lot more personal protective equipment for the doctors and isolating people a lot better, what else would have made this episode better, do you think?
So there was also this bit about the virus mutating into an airborne variant really quickly. Probably Deacon Ellick designed it to be airborne, because viruses don't just spontaneously mutate to gain new function within a day. It usually takes a lot longer for that to happen. It would have been, it takes a lot longer for viruses to gain new function, and they do change, but generally not in very predictable ways. And generally, they don't really become airborne. This is not a thing we see very often, actually.
That's a pretty
It's
major
either airborne
switch.
or not.
It's a pretty
Oh,
major
yeah.
switch. I was trying to think of the personal protective equipment. I think when Vedic Baral was really sick, I remember Bashir wearing some strange gown and hat. I don't remember if he had a mask on, but I remember a very strange outfit as he was operating on him.
Oh, yes, he had a weird outfit on. I think he might have worn a mask there.
He may have worn,
But
but you're right. It's very rare, though.
there's one other episode that's actually really good for that, and it's in Enterprise. It's in the 4th season of Enterprise, and it's called The Observer Effect. And that episode actually has a really good treatment of quarantine and personal protective equipment.
Good.
So go watch that.
I love Enterprise. I don't feel like it gets enough love.
That's actually a good episode.
Yeah,
I
one of
really
my
like that
favorites.
episode.
Any final comments on this one before we move on to the next episode?
Curing a virus is not a one-man mission. Don't leave it up to a single doctor that's been infected. Maybe tell someone about it.
It's a common TV problem in general.
Yeah.
All right, well, let's move on. Let's move back to Voyager again. I've been bugging you guys to give a lot of these explanations, so I'll do this one real quick. Let's talk about the Voyager Vidiian phage. So this came up starting early in season 1, but then actually it's something that spanned multiple seasons of Star Trek Voyager. So there's a species called the Vidiians. They'd been infected with some sort of virus that caused this decay of their organs over the last numbers of thousands of years. To fight the infection, they actually made great strides in medical engineering, which is awesome. However, their main approach for dealing with this is to graft tissue and organs from random alien species that they happen to come by and capture and replace their own tissues while they're succumbing to the phage. And it looks like then even the new organs start succumbing eventually. In the latter half of the first season, we learned that Klingon tissue or DNA, it's not clear, something about Klingons is actually resistant to the phage, and that's why they're very interested in B'Elanna Taurus. And it's kind of quiet for a long time aside from just some quick interactions, but in the 5th season we learned the Vians had been cured, though no specifics were given, except it was done through that think tank group, which had the actor Jason Alexander, who had played George Costanza in Seinfeld.
We all know think tanks are very effective.
Yeah. So with all of that, are there viruses like this that we know that affect humans that sort of decay of tissues?
So, not specifically. The Vidiian phage actually seems to cause a disease that resembles leprosy, or late stage treponemal infections like syphilis and yaws.
Again, don't Google that. Please don't Google that if you want to spare your eyes.
That's amazing. So the term phage, my understanding, and you can correct me, you're the virologist, is that comes from bacteriophage, which seems to imply that they infect bacteria.
Yeah, so bacteriophages infect bacteria specifically, but there are other phages that can infect viruses as well, virophages.
Okay.
Phages.
I didn't know that.
Yeah, and but we don't know of any phages that infect mammalian cells yet that I know of anyway. And I was thinking that perhaps one mechanism of how the disease caused by the phage could happen would be that the phage was destroying the Vidiian commensal microbiota. Like
that's a beautiful idea.
Yeah, infecting the commensal bacteria of, like, say, the Vidiian gut, and then the subsequent I guess what we would call perturbation, would then lead to disease by infection from other opportunistic pathogens, kind of like in the same way that we have quite damaging infections by Staph aureus and C. diff.
So in that sense,
Or
it's not actually attacking the cells of the Vidiians, but it's attacking the cells of the microbiome of the Vidiians, and that's what's harming them. That's beautiful.
exactly.
That's very creative.
Yeah.
I love it.
And I guess another explanation could be that perhaps that the phage was just kind of a common name that the Vidiians would give the pathogen causing the disease. An example of this would be in the early 20th century, late 19th century, there was rodent poison that was marketed as virus, but it was actually bacteria. It was salmonella. So the farmers were dosing fields with salmonella, trying to
A bad
kill
idea.
on the field mice.
Wow.
Yeah.
So my understanding is that that people now actually there's trials for phage therapy to actually try to help you in the sense of basically trying to knock out, for example, antibiotic resistant bacteria and things like that. So phage, if anything, are very helpful for us potentially.
Oh yeah,
Oh yeah,
yeah.
you can get this. Audrey, you want to speak about this?
The phage therapy.
Yeah.
Yeah, it is a thing. I think this is actually something that is what has historically been way more popular in Russia and the Soviet Union.
In Georgia, actually.
Oh yeah, in Georgia.
Not the
Russian
state.
Georgia.
Not,
Soviet Georgia, sorry.
yeah,
It's not
not the
Russian at
state.
all.
um, I think they had been working on that since the 50s. A big problem, though, with phage therapy is its potential unintended consequences, given that you're dealing with these biological organisms that can act in unpredictable ways.
Would
So
there be
it's
a
not,
direct threat, or do you think it would be like what you said before, where it might be attacking your microbiome, or both?
could be that too, yeah.
Yeah.
So it's a lot easier in a sense to design, I suppose, other sorts of antimicrobial therapies and that. But yes, phage therapy is a thing.
Yeah, if I can say something about
Please.
that, the great advantage of phage therapy is that it's specific. Antibiotics, which we use against bacteria, it's a very blunt instrument. It's a hammer. It kills everything. And you can design a phage that will kill only the bacteria you want to kill. So it can be really specific if it works. But we're not quite there yet.
Could it mutate and then broaden?
I think that's what Audrey was getting at.
That's
Saying
okay.
that,
I see. That's the point.
it could mutate afterwards.
Yeah.
And then maybe infect all of your microbiota, which you don't want at all.
No.
But in theory, you can design a phage that's going to kill only the problematic bacteria.
Cool.
Yeah.
Any final comments on the Vidiian phage?
Yeah, I also wanted to mention
Please.
that the Vidiians tell Janeway that the phage destroys their genetic codes and their cellular structures. And so many viruses can cause organ failure in the infected, but actually destroying parts of the genome is another issue entirely. I think with perhaps the exception of retroviruses and other what we call oncoviruses, cancer-causing viruses, you wouldn't have this sort of effect. And papillomavirus and Epstein-Barr virus actually encode for viral oncogenes, which purposefully induce uncontrolled cellular proliferation in the host, which is essentially what cancer is. So, and in general, it's characteristic for many cancers to have what can be described as destruction of the genome, like huge chromosomal rearrangements, deletions. The destruction of of organ systems in the infected individual would probably come from a cancer like pathology induced from the virus. And then from there, you can imagine how the Vidiians would need like skin grafts and organ transplants and so forth.
Awesome. Yeah,
So Mohammed, do you think this episode could be made better in some way, since you asked us this about all the other episodes?
That's a good question. I mean, honestly, I think the best idea is, and credit to Audrey because she just said it. I like the idea of it attacking the microbiome as opposed to attacking the actual cells of the host. At that point, then it really is a phage, and it's a lot more obvious how you could potentially cure it and things like that. So I think that actually works really well. So I mean, but credit to you, Audrey, for coming up with that solution. That's fantastic.
That also explains why it wouldn't infect the crew.
Exactly.
Why it doesn't infect any of the humans or Klingons, etc.
Exactly. Cool. Well, we have time, I think, to do one last one, at least briefly. Let's talk about Deep Space Nine: The Quickening. Does one of y'all want to give a quick description of that? We can be a little brief on that because we're running near the end of the hour now.
Yeah, so The Quickening, I like this episode because it is it's a slightly better treatment of an epidemic in the sense that it's not doesn't have a magic cure at the end, and it's a bit more realistic in that sense. And that's when Bashir and Dax and Kira go to the Gamma Quadrant, and they find a planet Teplin, which had been infected by the Teplin blight, which is a virus that was engineered by the Dominion because they didn't want to submit to the Dominion. And this virus causes lesions on all the people, and at some point it quickens and they die. And Bashir, of course, tries to cure it, but then fails. But in the end, he does manage to make a vaccine for it. So I don't want to speak too much about this episode because I think it's a bit of a downer, but because it's more realistic, I think it's a good episode to think about.
It was a sad one. So an interesting thing with that one was how the cure didn't affect the people who had it, but it affected their offspring. Is there anything that y'all know that's like that now where, basically you can become immune in the next generation to a virus? Some sort of maternal effect? I don't know, I'm just making that up.
So I don't know about anything about being immune afterwards. I mean, there are several viruses which are transmitted vertically, mother to child, and they do have some effects on the sex ratio of children being born.
Oh, true.
But I don't know too much about that.
I guess that's true because it's not. Is it? I don't think was it actually communicable or was it actually in their genome? I don't actually remember. Or did they say?
So it looked like it was. I'm not entirely sure because when the baby is born, it is without disease. Yeah. So what I was thinking is that it was probably not infected intrauterine, and it wasn't infected while she was pregnant, and because in that case the vaccine would not have helped. Yeah. Because it would have already been infected. So it should either be infected during the process of giving birth or shortly afterwards. I see. And I don't know. It's never actually said. It is said that the virus, that the physiology of the Teplins are too different to humans or Trill, that Dax or Bashir could be infected. Yeah. And that makes sense because, you need special receptors for the virus to enter your cells. Mm-hmm. But I'm not sure how this one was transmitted exactly. It's never actually shown being transmitted. That's true. That's true. It already has the blight and the scarring from that. Mm-hmm. It's definitely a sad episode. Yeah, but I think it is vertically transmitted because they were looking at the baby, looking for the scarring, looking for the lesions. It's true. So they were expecting it. Okay. Which means it should be mother to child.
Maybe it was a retrovirus.
It was a retrovirus.
Probably.
Everything's a retrovirus.
Of course, yeah. Yeah, maybe something I want to mention with this episode is that it does fall back onto this trope of having a virus suddenly starting to mutate a lot. And I think it's actually an unnecessary trope. It makes for a lot of tension in the episode and it's easy to explain it away. But you can actually make a lot of things happen by just gene regulation, by just triggering things without changing the genome. So
it's basically just changing which
think,
genes are turned on and which genes are turned off. That's what you mean by gene regulation.
oh yeah, that's I guess more of your work than mine. But yeah, you don't need to have intense mutation to have a sudden change happen.
Yeah, that makes sense.
You just environmentally change something and then suddenly you quicken.
We're getting near the end of the hour, so I guess we have to ask the question that I'm sure some people watching are thinking right now. There's obviously a certain virus on everybody's mind right now, and it's not the flu. COVID-19. What do you all think is like, what are the prospects? Any comments or anything you'd like to share with people watching this about that? I mean, I'm sure they'd love to hear from people who actually know viruses about this.
Well, Lucy is, he's actually working on COVID-19 and SARS-CoV-2 as a molecular epidemiologist. So I am so very interested to hear what you have to say.
So I have been looking at SARS-CoV-2. That's the virus that causes COVID-19. I've been looking at SARS-CoV-2 genome since January and doing a bunch of work on it. So the prospects is that it's not going away anytime soon. Sorry to be negative, but it's not. It's serious. It's going to be here for a while. If I should just throw out a couple of things, it's that we don't expect the virus to change significantly within a year or even 2 years. So this virus has, of course, acquired mutations since it first started spreading in humans. That's normal. All viruses acquire mutations. That always happens. Most of these mutations don't make any change to the function of the virus or to how sick people are going to get or how transmissible it is.
Or an efficacy of a vaccine.
Oh yeah, certainly not. People have done modeling on this and found that the spike protein, which is actually something which is in the SARS-CoV-2 virus, which is the target for most vaccines, it has not mutated enough to make a big change in whether or not a vaccine would work. But yeah, so the main thing is that it's not going to magically become, or not magically, but it's not going to become a lot more transmissible or less transmissible or a lot less severe over the course of a year. That's something that takes much longer. And there is this sort of conventional wisdom that viruses always mutate to become milder over time and sort of live along with their hosts. And that is actually wrong. I don't want to be negative again, but a virus can actually become more virulent. It can become more severe as time goes on, because a virus it's just affected by natural selection. It's not something which thinks. It's not something, we like to characterize it and say we're fighting the virus and the virus is sneaky and it's
like a computer virus, right? Same idea.
Oh yeah, it's a program. It reacts to stimuli, and yeah, it's not going to change in that way. I've kind of lost my thread now. Sorry. Yeah, I just wanted to mention those things that, and then I think I also want to mention that I don't think herd immunity is the way out. I think there are better things we can do. I think it's a risky strategy. And
when you're saying herd immunity, you're referring to herd immunity through community transmission, not herd immunity because everybody took a vaccine.
Oh yeah. So herd immunity through community transmission, I don't think is the way out.
Yeah.
Hopefully a vaccine is the way out. That would be the best case, I think. But yeah, it's a severe disease, even for young people, and it can have severe, long-lasting effects. Maybe, no, I don't think I want to talk too much about that, unless there's something specific you wanted me to answer.
We're just about out of time anyway, too, but that was really helpful. So I guess let's close out with the usual advice: mask, social distance, and then live long and prosper, too. Thanks again for you guys for doing this.
Long and prosper.
Enjoy the rest of the
Thanks
convention.
For talking to us. Thanks.